A recent viral headline claimed that a consumer AI chatbot independently cured a dog's terminal cancer. The actual subject is Rosie, a 7-year-old Shar-Pei. After a diagnosis of late-stage mast cell cancer, standard chemotherapy failed to stop her tumor growth, leaving her with a terminal prognosis. The owner utilized consumer artificial intelligence as a bioinformatic tool to architect a personalized mRNA vaccine. Messenger RNA, or mRNA, is a molecule carrying genetic instructions from DNA to the cellular machinery responsible for producing proteins. This distinction highlights a shift in medical research. These tools act as accelerators that can navigate complex biological data at speeds that were previously impossible for individual researchers. The workflow began with extracting Rosie's raw genomic data through next-generation sequencing, or NGS. NGS is a technology that allows for the massively parallel sequencing of entire genomes to identify specific genetic variations. An AI identified the necessary Illumina sequencing equipment, leading to the University of New South Wales Ramachadi Centre, where scientists performed the physical DNA extraction. Healthy tissue was sequenced at 30 times depth, while tumor tissue required 60 times depth to isolate mutations. AI filtered 320 gigabytes of data, narrowing massive genetic code to a single target, a c-KIT protein mutation. This created a neoepitope, a marker the immune system can recognize. In this phase, the AI processed the massive data library, while the physical generation of evidence relied entirely on university-grade laboratory infrastructure. The team attempted to visualize the mutated protein using Google's AlphaFold. AlphaFold is a deep learning system that predicts three-dimensional protein structures. The resulting model carried a confidence score of 54.55, which required manual validation by human structural biologists due to the risk of error. AI models translated the protein data into the mRNA open reading frame, which contains instructions for protein synthesis. Scientists at the UNSW RNA Institute then physically synthesized this mRNA, encapsulating it within a lipid nanoparticle, a fatty spherical carrier protecting the fragile mRNA during cellular delivery. This breakthrough relied on a clear division of labor. Algorithms functioned as the digital architect, while the university infrastructure served as the physical manufacturing facility. The custom LNP mRNA vaccine was co-administered with a checkpoint inhibitor. Checkpoint inhibitors are drugs that prevent cancer cells from switching off the immune response, allowing the vaccine to be more effective. This chart tracks the treatment's physical outcome. By mid-March, Rosie's primary tumor mass was reduced by 75%. Her mobility was restored within months, and she returned to chasing rabbits and jumping fences. The process required millions of dollars in existing laboratory infrastructure and continuous human clinical oversight. These results were achieved within a highly controlled academic partnership, rather than through independent at-home software. By democratizing bioinformatics, these tools allow researchers to compress the research and development timeline for personalized medicine. While regulatory and scaling barriers remain for human applications, this case demonstrates how AI can drastically shorten the path between diagnosis and a custom therapeutic.